Hello, Senior 2. Now we're going to explain a new lesson. Okay, our lesson today about Unit 3. Okay, Unit 3 consists of two lessons. First one is about respiration in living organism. Okay, respiration in living organism, can we explain it? First one in general, how what the difference between respiration and breathing, and second one respiration in human with the complete description about the respiratory system. Lesson 1, okay, about the cellular respiration. Cellular respiration, okay, it's a process by which the cell can get its need from oxygenated blood, oxygen gas from the oxygenated blood, okay, and release its carbon dioxide that get out from respiration. Okay, so the aim or the goal of cellular respiration is to break down the monosaccharides like glucose and get its energy, to release energy that used in stored in the shape of ATP. ATP is the currency of energy. Importance of glucose, carbohydrates, especially glucose, is considered as of stored energy. So here, carbohydrates or the glucose, okay, stored energy inside your body, because if your body need energy, so it will burn or oxidize the glucose inside the cell. Okay, that's meaning of cellular respiration. So cellular respiration is meaning by oxidation of the monosaccharides like glucose to release energy, and with releasing of energy, okay, water and the carbon dioxide are released, that can be transferred from one cell to another and from one living organism to another. The glucose molecule is considered as an excellent example to study the steps of breaking down the food molecules, as it is the molecule commonly used by the majority of living organisms to produce energy more than any molecule of available food. So here, a glucose molecule to us, okay, represent as ideal molecule for respiration. Mitochondria is the place or the home of respiration. Okay, mitochondria is organelle inside the cell, okay, a eukaryotic cell, and this home, okay, consists of two parts, outer part and inner part. Outer part is outer membrane, and as you see here we have inner membrane. The difference between outer and inner membrane is a smoothly structured, while the inner membrane is consists of folded convolutions, as you see here, to increase the surface area of cellular respiration. Okay, let's get inside the mitochondria deeply. Consists of matrix, and this matrix is like the ground of the mitochondria. Okay, the name of this folds is named by Christie, and matrix consists of ribosomes, okay, and granules, and also of DNA. DNA here, it's a function to form enzymes that help in cellular respiration. Okay, also we can found ATP, okay, particles or ATP molecules. Okay, this is the complete structure of the mitochondria. Mitochondria is a place where the cellular respiration takes place and energy produced and stored, okay, like photosynthesis process with the chloroplast, with the home of photosynthesis process and storage of starch and the glucose. Okay, the respiratory enzymes, water, coenzymes, and unilater meaning of coenzyme, co meaning a cooperative, so it helper to complete the process of cellular respiration. Phosphate group to build up ATP, electron carrier, and unilater known the function of electron carrier from its name, carry electrons. Okay, from chemistry you studied the electrons after excitation and can be excited and carry more its need of energy and turn to active electron, okay, and by losing its energy back to its stable state, and this energy can be stored inside ATP molecules later in need. The most important coenzymes, we have two types of coenzymes. First one, NAD, second one, FAD. NAD, reduced, meaning of reduced, react with hydrogen. Please here focus about meaning of reduction and oxidation. Reduction meaning, first one is react with hydrogen or gaining of electrons. So meaning of reduction, react with hydrogen like NAD and FAD. NAD react with hydrogen, reduced by hydrogen to NADH, and release one H because it's enough with only one proton or one hydrogen. Second one, FAD, reduced also with the hydrogen, and it's completed with two protons or two electrons, FAD plus H2, FAD H2 reduction. An energy required by a cell need ATP, which is considered as universal currency of the cell. And when ATP changes to ATP, an amount of energy is released. So here we play on the transformation of ATP to ATP for storage, or from ATP to ATP for releasing energy. Role played by ATP or adenosine triphosphate molecule. Any energy required by a cell needs ATP molecule. ATP molecule is considered as a small currency of the energy, so represented by the currency of energy. It can be easily spent and exchanged, can easily transferred through all over parts of the cells. It can be considered as the universal currency of energy inside the cell. The structure of ATP molecule. ATP molecule, its components inside the cell are present. ADP, adenosine diphosphate, and P phosphate. So ADP and ATP. ADP, first structure, adenine, which nitrogenous base. Adenine, you had it before in senior 1. Ribose is a penta sugar or 5 carbon sugar, pentose. Number 3 phosphate group because it's ATP. Those are linked together by two high energy bonds. During cellular reaction, only one of these bonds usually break down. Only one phosphate group is removed by hydrolysis or reaction with water of an ATP, which become ADP. ADP, adenosine diphosphate. One ATP, by measure can contain about 7 to 12 kilocalorie per mole is released. This is by molarity, while by the number or the amount of energy by number of molecules, one ATP consists about one molecule of glucose, consists about 38 ATP. Adenosine triphosphate, consists of adenosine diphosphate, one phosphate group, after oxidation, and energy that released about from 7 to 12 kilocalorie per mole. Steps of cellular respiration. Cellular respiration is occurred depend on its occurrence on 3 steps. This complete oxidation of glucose. Maybe the glucose not completed oxidized. But we here looking for the complete oxidation of glucose. The first step start at cytosol of the cell. Cytosol meaning of the cytoplasm, the empty cytoplasm from organelles inside the cell. This is the first step and named by glycolysis. And second and third step occurred inside mitochondria. As you see here in this equation, C6H12O6 is the symbol of glucose. By respiratory enzymes, complete oxidation turn to carbon dioxide 6 molecules, water 6 molecules, and 38 ATP. This is a complete oxidation of 1 molecule of glucose. Cellular respiration takes place in 3 major stages. First stage is glycolysis and take place inside non-organ part of the organelle part inside cytoplasm or cytosol of the cell. Krebs cycle, electron transport, all of them, okay, occur inside mitochondria where respiratory enzyme, water phosphate group coenzymes, and electron carrier molecule are exist. So oxygen here, we will know if it's important or not important and where it's important. Hydrogen carriers, okay, during glycolysis breakdown of glucose and the Krebs cycle, hydrogen atoms are removed from carbon skeleton of glucose molecule that pass coenzyme NAD and the fat. NAD and fat are coenzymes that carry the electrons in shape of hydrogen, okay, and turn into the last step, electron transport, to release its energy. NAD plus H2 turn to NADH as we said before. Fat plus H2 present fat H2 by reaction with hydrogen is named by reduction reaction. Okay, look here, glycolysis. Look like this first stage in cellular respiration. Glycolysis takes place in both aerobic and aerobic. Aerobic meaning with oxygen or the presence of oxygen. Anaerobic without presence of oxygen. Glycolysis, one molecule of glucose break down forming two molecules of pyruvic acid. Please look carefully here. Start with glucose 6 carbon, okay, and a little bit with change into glucose also 6 phosphate, meaning of glucose 6 phosphate, okay, that the carbon 6 will bind with one phosphate group, okay, so we need one phosphate, so change it from ATP to ADP. After, glucose will change also into fructose without a change in the number of phosphate, so here we don't need any molecule of ATP. After a little bit, fructose 6 will change to fructose 1,6. So here we have two positions for phosphate, so we need another phosphate group from ATP to ADP. After fructose 1,6 phosphate will break down into two phosphoglyceraldehyde. Okay, phosphoglyceraldehyde, same structure of phospho, and consists of except C6 to 3 carbon. Okay, so no change here in the number of carbon, no change here in number of phosphate. After two phosphoglyceraldehyde, going to change into the last one in this stage, pyruvic acid. Pyruvic acid also consists of carbon 3, but not hydrogen 6. Okay, hydrogen 4. So we have here two hydrogen will get out. Hydrogen carried by NAD to NADH. So we have here two hydrogen, two molecule of coenzymes to NAD to NADH. Also we have here two phosphate. Okay, and from each molecule 4 ADP plus 4 ATP. Okay, 4B will form 4 ATP. 4 ATP and you consumed before 2 ATP, so the total ATP that get out from this stage is 2 ATP after deleting the consumption. Pyruvic acid has two routes, aerobic or anaerobic. If get inside anaerobic, okay, maybe fermentation, okay, like acidic fermentation or alcoholic fermentation, okay, or they get in aerobic and get inside the Krebs cycle. Oxidation of glucose molecule into pyruvic acid is accompanied with the reduction of two molecules of NAD into two molecules of NADH, production of two molecules of ATP. So here produced two NAD and two ATP. All these reactions occurs in the absence of oxygen. So this process occurred in aerobic and anaerobic, okay? Both types of respiration. The energy that resulted not enough to perform all vital activities. So this energy, the amount of energy that to ATP is not enough to get the cell all properties, activities, so need more energy. Therefore, in the presence of oxygen, pyruvic acid molecule pass into mitochondria to produce more energy. This take place in two consecutive stages, Krebs cycle and electron transport. Here, one molecule of glucose, okay, after oxidized, okay, and break down into two molecules of C3H6O3, okay, this is phosphoglyceraldehyde. After a while, okay, turn to pyruvic acid C3H4O3 and release to ATP. Krebs cycle. Krebs cycle take place in two stages. Stage number one, each molecule of the 2-pyruvic acid, molecule 3 carbon, is oxidized into presence of coenzyme. This coenzyme is coenzyme A, acetyl coenzyme A, okay, into 2 carbon. 2 carbon is acetyl, okay, and carried by the coenzyme, cooperative enzyme, okay, A, that join Krebs cycle and get inside Krebs cycle. Okay, so we have here two things present. First one, releasing of 1 carbon from each one of pyruvic acid. So we have here 2 carbon, 2 molecule of carbon dioxide. Also carbon is combined with hydrogen, so we have here 2 molecules of NADH. 2 pyruvic acids, okay, plus 2 coenzymes turn to 2 acetyl coenzyme. Acetyl is C2, so we have here 2 carbon released by carbon dioxide in form of carbon dioxide. Also we have 2 hydrogen turn to NADH. Maybe protein, amino acids or fatty acids get inside Krebs cycle. Acetyl coenzyme A. Coenzyme A is cooperative enzyme that get acetyl inside Krebs cycle. Look here carefully. Start of the Krebs cycle with oxaloacetic acid is 4 carbon and bind with 1 molecule of acetyl 2 carbon to form citric acid. After this combination and get the acetyl inside the Krebs cycle, coenzyme, okay, will get out from the Krebs cycle to get another molecule of acetyl to get inside Krebs cycle. Citric acid is 6 carbon, so the first result from Krebs cycle is citric acid, so the Krebs cycle named by another name is citric acid. 6 carbon will turn to 5 carbon ketoglutaric acid. 5 carbon, so here we have here reduction in number of carbon, so releasing in form of carbon dioxide. Also we have here oxidation in citric acid because of releasing hydrogen, so 1 molecule of hydrogen will get out by form NAD to NADH. Second step, ketoglutaric acid, 5 carbon change to 5 carbon succinic acid, succinic 5 carbon, so we have here 1 loose in carbon turn to carbon dioxide. Also H carried by the carrier of hydrogen NAD to NADH. 4 carbon also change to 4 carbon, but here, okay, we have not losing in carbon dioxide, not carbon, same carbon in number, but hydrogen will get out, okay, so FAD change to FAD H2 and ADP change to ATP. This is the only 1 molecule, okay, inside the first Krebs cycle. Succinic change to malic, and the malic change also to oxaloacetic. From 4 to 4, no change in the carbon skeleton number, okay, while the change also only here in the number of hydrogen from NAD to NAD. Please here focus about the number of NAD, okay, 1, 2, 3, okay, 3 NAD. This is Krebs cycle first one, okay, and you have 2 acetyl, so we have 2 Krebs, 6 NAD total. Number of carbon dioxide 1, 2, the total 4. FAD 1 in total 2, ATP 1 in total 2. 2 pyruvic acid plus 2 coenzyme, okay, get 2 NAD, okay, and 2 acetyl coenzyme plus 2 carbon dioxide. This is very important note. Acetyl group from breaking down fat molecule or protein molecule can combine with coenzyme to join Krebs cycle, as we said before. Stage 2. Each molecule of acetyl coenzyme join Krebs cycle, where its coenzyme split off to repeat its role. At the same time, acetyl group 2 carbon combine with oxaloacetic 4 carbon to form citric acid 6 carbon. Citric acid passes through 3 intermediate compound to form oxaloacetic again. These compounds are ketoglutarate 5 carbon, succinic 4 carbon, malic acid 4 carbon also. In the pathway of Krebs cycle, 2 molecules of carbon dioxide are released, 1 molecule of ADP, 3 molecules of NADH, and 1 molecule of FADH2. And Krebs cycle repeated twice, so multiply this number in 2. Number 3, electron transport chain. At the end of Krebs cycle, hydrogen with high energy electrons carried by NAD and FAD are transported by a sequence of coenzymes called cytochromes. Cytochromes are present in the Christie electron carrier present at the inner membrane of the mitochondria. These cytochromes carry the electrons, as you see here, like a ladder. Okay, hydrogen is split into 2 protons and 2 electrons. Each electron, okay, carry a huge amount of energy, and to release this energy you can split and move the electron downward on this ladder, okay, and hydrogen will weight the electron under this ladder, okay, to receive electron after releasing its energy. During this energy is released from ADP phosphate, okay, so here this energy change to or used in changing ADP to ATP. This is meaning of oxidative phosphorylation. This is the total amount of energy, okay. We have 2 NADH from glycolysis. So NAD, okay, consists of 1 NAD consists of 3 ATP, 1 fat consists of 2 ATP. 2 times 3 equals 6 from glycolysis. 2 NAD intermediate, don't forget, okay, between glycolysis and the Krebs cycle we have 2 NAD, okay, also 6 ATP, 6 NAD as 2 Krebs cycles plus 3 18, and 2 fat times 2, okay, as 2 Krebs cycle equal 4, so the total is 34. And we have also 2 ATP and in Krebs cycle 36, and also we have 2 ATP in glycolysis, so the total number is 38. Oxidative phosphorylation. At the end of Krebs cycle, hydrogen with high energy electron carried by NAD and fat are transported by a sequence of coenzymes called cytochromes, and the cytochromes is present inside the inner membrane of the mitochondria or the Christie. These cytochromes carry electrons at different energy level. These high energy electrons are passed from one cytochrome to another like ladder, at the same time by descend from higher energy level to lower one. During this, okay, the form of ADP is formed, transform ATP. This is meaning of oxidative phosphorylation. ADP plus B plus energy change to ATP. Oxygen is considered the last receptor, so it's a function only of oxygen to receive white hydrogen after be stable, okay, to form water. Electron with hydrogen, electron here after get down from cytochromes with half molecule of oxygen get 1 molecule of water. In the electron transport chain, each NAD release energy about 3 ATP, while 1 molecule of fat consists of 2 ATP. Accordingly, during aerobic respiration, each molecule of glucose produces 38 ATP, 2 of which are produced in the cytoplasm of cell during glycolysis. So in glycolysis we have only 2 ATP, and 36 ATP molecules are present inside mitochondria, okay, as transport chain of the electrons. anaerobic respiration or fermentation. This process occurs after after glycolysis, after formation of pyruvic acid, get inside human in some parts like muscles, also get inside bacteria, also happen inside fungi like yeast. The process by which living organisms obtain energy from food molecule in absence of lack of oxygen by help of special enzymes, and these produce small amount of energy, you said before, to ATP only. Glucose, glycolysis to pyruvic acid. Fermentation may be acidic, may be alcoholic. Fermentation acidic occur inside the cell, like muscles inside the human body or inside bacteria, and form two molecules of lactic acid, while alcoholic one that present inside yeast, or the plant cell, and form two molecules of ethyl alcohol and release two molecules of carbon dioxide. When oxygen is missing or in low quantity, living organisms are as bacteria and fungi respire by anaerobic respiration. That's not meaning that only respire anaerobic, this is in unfavorable time, but in favorable time, respire as usual aerobic. Some plant and animals may also respire anaerobic when oxygen is not available. This is also called by fermentation, and it doesn't need oxygen, but it takes place in the presence of some special enzyme. Anaerobic respiration begins with the same beginning of aerobic respiration, glycolysis. The glucose molecule is composed into two molecules of pyruvic acid, as you see before in glycolysis, with the formation of two molecules of NADH and a small quantity of energy to ATP. Pyruvic acid is converted according to the type of the cell in which it was formed. In case of animal, acidic fermentation, this is when the muscle exert a lot of effort, and the blood cannot reach with a huge amount of oxygen, so to get the muscle energy, a quick energy, not enough but a quick energy, so it will turn the pyruvic acid to lactic acid to get a small amount of energy. This is meaning of muscle fatigue, or the pain that occur inside the muscle, as a result of the formation of pyruvic acid to lactic acid to release a less amount of energy till a huge amount of oxygen can reach to the muscles. This is known as muscular fatigue. If oxygen is available, lactic acid is converted into pyruvic acid again and then into acetyl coenzyme, so it will turn from anaerobic to aerobic. In case of bacteria, pyruvic and acid also change into lactic acid, the meaning of acidic fermentation. In case of bacteria, pyruvic acid converted into lactic acid, as you see here. In case of yeast, fungus, or in some plant, is named by alcoholic fermentation. So pyruvic acid here change into ethyl alcohol and carbon dioxide. This is used in a lot of industries, like bakery industries. Thank you and goodbye.